del Departamento de Farmacia. Estamos haciendo algunas pruebas técnicas de audio y video. Sean bienvenidos. El día de hoy tendremos como ponente en el seminario a la doctora Thais Gratieri. Ella está conectada vía Zoom desde Brasilia, en Brasil. Su ponencia será en inglés y está titulada como Desarrollo de formulaciones para administración vía tópica y métodos alternativos in vitro para el análisis de medicamentos. Primero vamos a presentar una breve semblanza de la doctora Thais Gratieri en inglés, después hacemos un resumen en español, posteriormente pasamos a presenciar su ponencia y finalmente tendremos una sección de comentarios y preguntas. Si alguien desea externar al final algún comentario o pregunta en inglés, lo puede hacer así, o si quisiera en español, nosotros hacemos una traducción.
Associate Professor at the University of Brasilia and Substitute Coordinator of the Postgraduate Program in Pharmaceutical Sciences. She has a degree and a PhD in Pharmacy Biochemistry from the University of São Paulo Ribeirão Preto. She carried out a doctoral internship at the University of Saarland, Germany, and a postdoctorate at the University of Geneva, Switzerland, institutions which she still maintains scientific collaborations. She has experience in the field of pharmaceutical technology and cosmetology, with an emphasis on the development and evaluation of innovative systems for topical drug delivery. She has been developing several works focused on evaluating the performance of topical, ocular, cutaneous, nail, and vaginal formulations, including micro and nanoparticulate systems, through in vitro and ex vivo experimental models as alternatives to the use of animals. She is one of the founders and coordinators of the Cosmetic and Pharmaceutical Technology Laboratory, where, in addition to academic research, she works on technological product development projects in collaboration with pharmaceutical and cosmetic industries. Elected affiliate member 2022 to 2026 of the Brazilian Academy of Sciences, in 2012 she received the CAPES Thesis Award, and in 2013 the For Women in Science Award from UNESCO, L'Oréal, and the Brazilian Academy of Sciences.
Muy bien, como ya presenciamos, la doctora Thais Gratieri es actualmente profesora asociada en la Universidad de Brasilia. Ella actualmente es coordinadora de un programa de posgrado en ciencias farmacéuticas. Tiene el grado en doctorado en farmacia y bioquímica por la Universidad de São Paulo y llevó a cabo una estancia doctoral en la Universidad de Saarland en Alemania, también en la Universidad de Ginebra en Suiza. De hecho, pudimos coincidir en algún momento allá en la Universidad de Ginebra, en Suiza, ambas instituciones con las cuales mantiene actualmente una colaboración muy cercana. Ella tiene experiencia en el área de tecnología farmacéutica y cosmetología también, con un énfasis en el desarrollo y evaluación de sistemas innovadores para liberación tópica de fármacos, que es precisamente el tema de ponencia del día de hoy. La doctora Thais Gratieri ha desarrollado diversos trabajos enfocados en evaluar el desempeño de formulaciones tópicas, oculares, cutáneas, también como permeación a través de uñas, así como para aplicación vaginal, incluyendo micro y nanopartículas, a través de modelos ex vivo e in vitro como alternativas para el uso de animales. De hecho, la doctora Thais Gratieri es pionera a nivel mundial en la exploración de nuevos métodos que evitan el uso actual de modelos animales. La doctora Thais Gratieri es una de las fundadoras y coordinadoras de lo que se conoce como Laboratorio de Tecnología Farmacéutica y Cosmética, LTMAC, allá en Brasilia, en donde además de llevar a cabo actividades de investigación, trabaja la doctora Thais Gratieri en el desarrollo de proyectos tecnológicos en colaboración con industrias farmacéuticas y cosméticas. Ella fue elegida como miembro de la Academia Brasileña de Ciencias y en el 2012 recibió el premio a la mejor tesis, y en el 2013 la doctora Thais Gratieri recibió el premio para mujeres en la ciencia por parte de la UNESCO, L'Oréal y la Academia Brasileña de Ciencias. Así que damos la bienvenida a la doctora Thais Gratieri. Doctora Thais Gratteri, please, very welcome to our meeting. Thanks a lot for your time, and you can start now.
Hello, everybody. Yes, can you see the presentation? Yeah? My internet just dropped while you were talking, but okay. Well, thank you very much, Professor Gerardo, for the invitation. And thank you all for being here. It's a great pleasure to share some part of our work with you. And today I'll talk about formulation development for topical drug delivery and in vitro alternative methods for performance analysis. First, well, I know Professor Gerardo already presented me, but I graduated in University of São Paulo, and then I spent some time abroad in Germany and Switzerland, where I met Professor Gerardo. Great pleasure. And since then, I've been professor here at the University of Brasilia. And this is part of our group. These are my dear students, some of them. And in our lab, it's called LTMAC. And if you want to look it online, feel free to visit our website. It's ltmac.com.br. And we are 3 coordinators, and we have now about 40 students. And for those of you who are not aware of Brasilia, is the capital of Brazil. And it's a city that was a planned city. It was completely planned. There was no one here. Then they planned this to be the capital of Brazil, and it was built in 1960. So it's a new town. And it was planned in this form of an airplane, as you see in the picture. And it's a very dry region. We're in this Cerrado region. So they also planned this lake, this huge lake. It was artificial at first, and now it's natural. And here are some iconic pictures of Brasilia. It has some famous architecture monuments. So when you're planning a trip to Brazil, please include Brasilia in your itinerary. It's an interesting city to visit. And for those of you who work with pharmacy technology and nanotechnology and want to collaborate with us, I will present some part of our facilities. We have this is our lab in the university, and we have, full infrastructure for complete development and characterization of nanosystems. So we have this nanospray and extruder, manual extruders, atomizer, Beckham coulter. So we're equipped for this nanoparticle development, and also for the drug release and permeation experiments. So we have these Franz type diffusion cells, which I'll show some examples of work we've been doing with it. But some cells we also design them for some specific needs and some specific performance analysis. Sometimes we print some of these cells with using 3D printer, so we can have specific use of them. I will also have examples of that. And now we just acquired this Phoenix dry heat diffusion system, which is also very nice. And of course the Analytic Central. So I will focus now on the main things of my line of research. As I said before, we are 3 coordinators, so we have other professors working with 3D printing of pharmaceutical dosage forms and some solid forms for oral use. But my lines of research are now focused on development of topical drug delivery systems, which includes topical for skin, cutaneous use, ocular use, vaginal, and sometimes buccal. But I'll have examples of them. And as a consequence of this focus of research, we're also working with developing some in vitro models for analysis performance. So we can analyze the performance of these topical materials that we have developed. So I don't know if all of you are in the pharmaceutical area, and just being very simplistic, why topical? What's the advantages of using topical drug delivery forms? Well, and I will illustrate it, why topical, using some examples of the lines of research we use. For example, treating alopecia. Well, to treat alopecia, you need certain drug concentrations at the site of action. So you need certain amount of drug in the hair follicles. If you have an oral intake of this medicine, you have some effective medicine has to distribute for all your body to have a specific action in the follicles, but you have the drug in other places. With this, you have adverse effects. So the local treatment would be would be ideal to have a local action just where you want it. But there are numerous challenges, and challenges come from pharmaceutical ones, as drug solubility, formulation. Formulation must not run off. You want a formulation that you apply and stay there, and possibly ideally control the drug release. So you have some controlled amount of drug being released during all day. So you just apply the formulation once a day. You don't want to apply things to your hair twice a day. And then you have your hair looking dirty or sticky or with some not ideal sensorial aspect. So you have to increase drug solubility. You have to guarantee stability. But it has also to be a formulation that has a good sensorial feeling for the patient, so the patient won't use. So this is directly correlated with the acceptance of the formulation and, of course, efficacy. But, of course, the greatest challenge of all is the cutaneous barrier. The drug to have an action in the hair follicle, it has to cross the skin and be in the hair follicle. So all our barriers, we're talking about a topic application, so we're talking about external barriers, our tissues. Our tissues are made for protection. Their physiological action is to protect our body from the entrance of stuff, and as in the case of skin, also for the skin is specialized in protecting us from losing water from our body to the environment. And it has a very specialized barrier that is the stratum corneum, which is the outermost barrier of our skin. So it's lipidic. You have the dead cells all conveyed in lipidic structure, so the skin won't lose water. Water is a very small molecule. So if the skin is specialized in protecting us from losing water, it also protects us from entrance of other things. So the entrance of molecules is a great challenge. And we want molecules not only to, sometimes not only to penetrate the skin, but to permeate the skin and reach local vessels and to have some local distribution. So that's a real challenge. And for this, well, nanotechnology is used, is almost a revolution in the way we deal with formulations and drug molecules, because it has many properties, many desirable properties. First, it can increase drug solubility in a desired formulation, depending on the molecules and substances you use for assembling this nanosystem. And it can also control the release. you can also have a lot of drug there, and it's released slowly, and it can facilitate this once a day application. But the main advantage for skin and for some types of skin treatments is to increase the amount of drug that penetrates the skin. But here comes a catch. It's not, some people think, well, it's easy then. Nano is very small. That's why it's called nano. It's very small. It's nanometric. So it penetrates more. And that's not true. And that's a huge mistake, because you think one nanosystem, it has many drug molecules. And the drug molecules, let's say it's this size. So you have many molecules. So a nanoparticle, compared to the size of a molecule, it's huge. So you have huge systems compared to the size of a molecule. So you have a nanosystem. The spaces between each cell, you can't have a nanoparticle penetrating that. So how does it increase the amount of drugs that penetrates through skin? So you have multiple mechanisms for this. Some types of nanoparticles, as lipidic nanoparticles, they can form a film in the skin, like a film layer, and because they are so small, they don't leave many spaces between them. So they form this occlusive barrier. And this barrier, the nanoparticles form sometimes, they impede the skin from losing water. And this already hydrates the skin more and facilitates drugs permeation. So sometimes it's not the nanoparticle itself that is entering the skin, but it's causing some mechanism that facilitates drug to penetrate the skin. In the case of hair follicles, we have this phenomenon that nanoparticles naturally accumulate in the hair follicles because of their size. And there are some, depending on their size, they may accumulate at the superior region or the inferior region of the hair follicle, but naturally they tend to accumulate in the hair follicles. And even for the movement, the hair follicles make, well, not in the hand, but in the head, when you think you have many, many hair follicles, you facilitate this interaction. And once they are there, they can control the amount of drug that is being released. So we have explored a lot these nanoparticle systems for drug delivery to many diseases in which the hair follicle is the target for the pharmacological treatment. For example, alopecia, of course, and this we have explored using some nanostructured lipid carriers, for example, loaded with an association of drugs. So when you associate latanoprost and minoxidil, they have some complementary mechanisms of action. So you can imagine you have an enhanced performance. And this week, we have some examples of these types of nanoparticles. And an important thing in these studies is that we perform these permeation experiments using Franz diffusion cell, but in the end, you need to see the amount of drug that is really in the hair follicle and to relate it with the amount of drugs in the other structures. And this we also call the enhancement factor, or the targeting factor of the nanoparticles. So it's also important. And this we will use this concept of targeting the hair follicle also to evaluate different types of applications. For example, if we just apply a formulation there, or if you apply and you perform some kind of massage for some time, 5 minutes or 3 minutes, and we could see that when you apply this massage, this physical external force, this movement that you inflict on the hair follicles may facilitate the nanoparticle accumulation in these structures. Then you can see here in this graph that after 6 hours with the massage, we had a higher follicular targeting than when we didn't apply this massage, and also a higher follicular targeting when you compare to a solution. This black column is just the drug solution. And we have used this with different drugs as well. Here I have examples of dutasteride, of finasteride. Finasteride is a well-known drug used for treatment of alopecia orally, and they had many adverse effects. So a topical delivery would be ideal. But also, formulation intended for some applications, it's important that it doesn't run off. It's like doesn't drip. So, for example, when we were developing this formulation with minoxidil for the hair growth of the eyebrow region, this region. So when you apply this, it's important that the formulation stays there. And this conventional system for performing the permeation, well, it's called vertical transdiffusion cell. That's why there is some confusion, because it stands up. So it's called vertical transdiffusion cell, but the skin is applied horizontally, and the skin is horizontal, and you apply the formulation on top. Then after some time, you see how much of the drug penetrated the skin. But with this, you can see how if you have a formulation that runs off and a formulation that doesn't run off, it's probably that they will have the same behavior in this type of experiment. Well, you could do a very complete rheological characterization of the formulation, but for an industry, well, it involves having, you need a rheometer, you have specialized analysts to evaluate this formulation. Sometimes that's another step. And you need expensive equipment, and sometimes you don't have that available. So if you have a in vitro experiment that could also see the performance of this formulation, it would be ideal and very straightforward. So for this, it's an example that we printed these specific cell types. They are rectangular here because, well, we evaluated the performance of the formulation in this vertical position. But for this, we had 3 sections, and we applied the formulation in the middle one, and then we removed this grid and put the cell in the vertical position, and we quantified the drug in all the regions, so we could say really compare the formulations, the ones that runs off and the one that doesn't, and what would have the greatest performance, for example. And sometimes we also need these models, and here's that second part that I said, that we're interested in also developing in vitro models that we can analyze the formulations and see the performance of them. For example, when developing a nanoparticle to treat acne or some other follicular disease that is related with an increased sebaceous content of the skin. For example, the acne, you have some sebaceous increased release. So it's not, when you use a healthy skin, and okay, nanoparticles accumulate there, but how would the behavior be when this skin would have an increased sebaceous content, would be very oily? And so we developed also this model when we applied this specific type of oily layer, so we can simulate this oily skin. And the behavior of the nanoparticles vary a lot. And we can see that some nanoparticle would still accumulate in the hair follicles and some would not, because of this difference in the skin. Well, another line of research we have is also to work with natural products. And natural products have additional challenges for pharmaceutical technology. Well, you may be aware of this, but main challenge is stability. It's very hard to maintain a natural extract or an essential oil stability in a pharmaceutical formulation. And I know Professor Gerardo has some amazing work with some natural products. And here we have some partnerships with another group that identifies these potential fruits and these potential extracts from the Amazonia region, and we evaluate which types of actions they would have. And we have come up with some interesting actions. For example, this fruit is common in the Amazonia region. It's called cuchich, and it's very yellow fruit, and its extract we evaluated in fibroblasts in some culture cells in melanocytes culture, and we saw that they have depigmentation potential. Then, so they could depigment the skin and use to treat some of these cutaneous spots. And so we used this in a microemulsion formulation, and we evaluated also in an in vitro model. For this, we counted with some collaborations, and we evaluated in 3D printed human skin in vitro model. So they had cells donated from humans, and they cultured it in the lab, and they had the 3D skin with melanocytes, and we compared the effect with some formulation that is available for purchase, and this formulation was better than the one already commercialized. So it's an interesting use. And we also evaluated the potential use of the fruit, the extract of the fruit cupuaçu. I don't know if you know this fruit, but cupuaçu is used to make several drinks and sweets. It's delicious fruit from the north region, and we evaluated it also in cell cultures, and we could see it could be used for regenerative potential, so to treat skin burns, for example. And when we look at the genes, the extract act, we could see some improvement in the elastin gene. So we imagine this skin regenerative potential would also come without formations of severe scar. So also having some cosmetic potential. And for this study, we needed some model to evaluate. It was very hard to stabilize, to make this formulation stable. If we just take the extract, it's toxic. So if we take the extract and put it in a conventional cream, it's not stable. So we used a nanoparticle. So we wanted to compare some two types of nanoparticles, but we wanted to compare also the penetration of these actives. And for this, if you're thinking to use this, if in a burnt skin, we need some burnt skin model, because it's not enough to just compare these two nanoparticles with intact skin. I needed to some resemblance with this burnt condition. So we also developed this burnt skin model in vitro, and just burning the skin for specific time, some specific condition, and making histology cuts, and just to see which would be the specific conditions to simulate a first degree burn, a second degree burn, and so on. And another permeation of these in vitro models is the vaginal model. So it follows the same principle. When you have some cream, for example, with an antifungal to treat some fungal infection in the vagina, well, you need a formulation that ideally won't leak, that will stay there for a prolonged time. And so, okay, I developed a formulation with some mucoadhesive polymers to make this formulation not to leak. But how would I compare this performance with the other one? That is a conventional formulation. So using this, you can see the E photo. I don't know if you see my mouse when I point, but when you use this horizontally, you can see the performance of the formulations. So for this, we developed this vertical model when we take the entire vagina of the porcine. And here, please, when we use these tissues, we use the porcine, the pig, the vagina, pig skin, pig eye, because they're the ones that they're the tissues that are most similar to the human ones. And we take this from the abattoir, so it's the meat that would be used for consumption. And we take the entire vagina and we hang there, so we have the vertical position, then we apply both formulations and you can compare the performance of the formulations. And here, just some example, we have all these columns comparing formulations in the horizontal position and then at the vertical position. Well, I don't need to say much about each formulation, but you can see that when you use the horizontal position, they all look more like the same. You have some differences from one group to another, but inside each group, I have these groups of 3 types of formulations with different concentrations, but they're all the same. But when you see they're vertically, you can see a very clear difference in performance. So you can see which formulation would be better than the other one for this purpose. So that's the importance of the in vitro model. And I don't have to say that it's much easier to perform an in vitro experiment than to use a live animal experiment or to perform a clinical study using humans. So when you can, like this, select the best formulation, then you can proceed with just this formulation. You take 2 or 3, you don't need to take 9 for a clinical study, for example. And at last, I'll talk a little bit about the eye. And when we think about topical formulations for the eye, for the ocular delivery, we also have these challenges of protection. So the eye is a very specialized organ of our body that is also protects us from foreign substances. And the first challenge is the volume that the eye supports. So you can put, for example, 1 drop of something in your eye that will stay there, about 25 to 30 microliters. If you apply 2 drops, the first time you blink, you have the excess running out. So you lose the formulation. So that's the first challenge there, the small volume you can apply. And the second challenge is once you apply something, you have a natural lacrimation, so you're forming tears in your eyes. But when you apply something, you have this more pronounced. So you have this tear formation and you wash the formulation out of your eye. So you have this draining of the formulation and it's drained to the nasolacrimal drainage here. So you lose the formulation. And the main technologies to increase bioavailability of something topically applied is something that makes the formulation to stay in the surface for a prolonged time. So you call this prolonged residence time. So it stays there longer, so it can better interact with the tissue and then penetrate more. And, well, when you're developing some formulation, you also have to compare these formulations. So we have these in vitro models as well. You can use the excised cornea tissue when you're thinking about something to treat some infection of the cornea, or you can use the whole globe, the whole eye globe. It's now becoming more common. But you don't have any of these defense mechanisms. You don't have lacrimation. You don't have the blinking. So we're now developing some models in which we use the porcine ocular globe, but we also apply some flow, some lacrimal flow, simulating the tears that will wash this formulation. And once again, when we compare these different models with the simulated tear and without, so without, we cannot differentiate very much the formulations, but when we apply the tears, we can see that some group of the formulations clearly stands out of the other ones, and you can say which group of formulations are better than the other ones. But you still need porcine eyes, and they're hard to obtain as well. For an industry, it would be very difficult to collect the eyes and transport and perform these experiments. To preserve these ocular barriers, you need to use the tissue at the same time, the same day they are excised, so you cannot freeze the eyes to use another day, because then the barrier will not be the same. And it's very complicated. So now we are developing a complete in vitro model. We're trying to synthesize a polymeric film that simulates the surface of the cornea, so we can evaluate the residence time of formulation. And in this in vitro model, we have the simulated tear flow, and also we want to apply a probe that will move on top of the formulation to simulate this blinking effect. And as this film is completely synthetic, it would be easy for an industry or any lab to use and to compare the residence time of formulation. And here I have some picture of this prototype. We're still developing, we're still trying different designs, but we're working with it now. So these were some examples of some things we've been doing so far. And here are my contacts. Feel free to contact. And if you have any questions, I'll be happy to answer them. Thank you.
Dr. Thais Gratieri, thanks a lot for this interesting presentation. Now we open our session of comments, questions. Somebody has a question or comment?
Thank you very much for your presentation, Doctor. I have a question about your in vitro models for skin. I mean, how do they work? You said almost every one of them were histological cuts, aren't they?
I couldn't hear.
All your in vitro models for skin, are they histological cuts? All of them? Aren't there any artificial tissues?
Okay. Well, yes, there's some artificial, the industry has tried some completely artificial layers to simulate the skin, but until today there are no artificial tissues that completely simulate all the skin layers and that has such an effective barrier for penetration. And even these artificial layers, I think they're used more for quality control or something like this in industry, because you cannot predict what will be the interaction in all the layers and how the drug will distribute, because it's a very complex layer with multiple layers. You have the stratum corneum, you have the hair follicles, and then you have the epidermis with some different lipophilicity. And the excipients of the formulation, they may interact with the stratum corneum and facilitate or difficult the penetration, the diffusion of a molecule. So it's not enough to just look at molecule solubility. You can look at solubility, you can look at the lipophilicity of a molecule and molecular size, but it's not enough. So if you change something in the formulation, you change the drug distribution. So it's very hard for these artificial barriers to simulate it. And you do have, I even have an example of this 3D tissue model. So you have stratum corneum, you have epidermis, part of the dermis, you can have this in vitro culture. And these skin models in vitro, they're used for studies of sensitization. So if something is irritant or not to the skin. But because the stratum corneum barrier, even though they're there, they're not as tough as in the actual skin and the actual excised skin. So they tend to, when you analyze penetration using this model, they tend to overestimate the amount that you would have. So until today, there's no substitute for the skin tissue in terms of skin penetration.
Then I have another question. How do you maintain those tissues alive? I think that's another challenge, isn't it?
To what? How do I what?
How do you maintain those histological tissues alive then?
Okay, yeah. Well, the skin you don't have to maintain alive. That's the main advantage of working with skin. And sometimes I tend to think that we are more evolved technologically in formulations for cutaneous penetration and transdermal penetration than to the ocular delivery, for example. I tend to believe because it's easier to work with skin because you can collect the skin and you can freeze it. You don't need to freeze it to -80. You just freeze it in a normal freezer, -10, -30, for up to a month. And you have this barrier maintained because the stratum corneum, which is the main barrier, is a dead barrier. The cells are dead and you have this lipidic nature. So if you dry, it's very important that when you freeze the skin, it's completely dried, because otherwise you can have crystal formation and then it can damage the skin. But if you maintain it very clean and dry, you can freeze it and use up to a month. So it's very easy. And the other ones, the vagina and the ocular, you need to use at the same day. So we have evaluated the barrier and how many hours it stands to transport. So we don't even need to maintain it in liquid, but just in the natural fluids it comes with. And you can use it up to a day at the same day. There's no problem, but we don't maintain them for longer times.
Thank you very much.
¿Alguien más tiene algún comentario o pregunta?
Hi, thank you for your presentation. It was excellent. It was perfect. And You already mentioned some of the problems and troubles you have with these all kind of formulations. And my question is, what are those main troubles when you work with the in vitro models or when you're working with the ophthalmics? And you also mentioned about the natural products. How did you manage to overcome these obstacles when working with the natural products?
The first question was, I'm sorry, it's hard to hear the question. The first one was what is the challenge with the in vitro models?
Yeah, what's like the main problems and troubles and challenges with working with these in vitro models, and also with the ophthalmic formulation? What's like the main challenges you have found with your prototype you are designing?
Okay, well, for the in vitro models, the main challenge is you want a model that has the minimal variability from one to another. So it's a practical challenge, because if you have some variation, for example, the tissues vary a little bit. If you use one skin, we remove the skin from the pig ear. So if you use a ear from just the one pig, you may reduce this variability. But that's not practical. You need multiple pigs. So you also have this inter-individual variability. But you need the model to reduce this variability to a minimum as possible. So when we are developing this, they have to have exactly the same dimensions. So you don't have errors, experimental errors, and that's a great challenge. You need the exact same dimensions. You don't need, you can't have bubbles in the receptor, and you can have temperature fluctuations. You try to control the temperature to a maximum as possible. Well, and we have this, I showed this, some apparatus. We have this Phoenix dry heat apparatus, which is specifically designed for cutaneous penetration. So it's a validated apparatus with this very specific measurements and so on, so you can reduce the errors. And when you have very reduced errors, experimental errors from one to another, you can compare formulations and you can see which one is actually statistically different from another one, and not just some variable you included. And when designing some performance, not I want to analyze something that is not this, so you don't have this complete validation. So that's a huge challenge, to maintain this, to be very strict when you perform the experiment, so you have minimal errors. That's a challenge. And for the natural products, the main challenge is the stability, undoubtedly. And many actives, like they come from, of course, you have some line of development. Well, first we evaluate the nectar extract or the essential oil. And when you evaluate the essential oil, you're talking about a mixture of substances. Of course, an involvement of this, a natural progress, would be to identify specifically which are the substances causing that action, and or maybe which substance positively interacts with the other one, so you can synthesize and have the substance synthetic in the future for this specific use. That would be the progress of this. But until you don't have that, it's good to work with the nectar extract itself. And for essential oils, for example, they are volatile substances. So to maintain their substances and stability, and it's very, very hard. So with some types of nanoparticles, we could enhance stability, and that's a great challenge.
Okay, thank you so much.
I don't know if I answered.
Yeah, thank you.
¿Alguien más tiene pregunta o comentario?
Hello, thank you for your presentation. I have a question. In your experience, it's absolutely necessary the experimental, the animal experimentation.
I couldn't hear.
Ah.
I'm sorry. When you read,
In your experience, it's necessary the animal experimentation.
Gerardo, can you please help me?
Hello.
I-
In your experience, is it's necessary the animal experimentation?
In my experience, and then it was the internet. I think I can't hear. It's not that I don't understand what you're saying, it's that I don't hear.
It's necessary to animal experimentation with you work with the skin and teeth model.
It's necessary to what?
To animal experimentation. Experiment.
Can you hear me, Thais?
Yes, now I can.
It's good. Thank you. The question is about if, in your experience, is the animal model is completely necessary, or you can supply with in vitro or ex vivo models. What about the trends, in your opinion?
So the question is if it's necessary
If it is,
to compare the in vitro and ex vivo?
oh no, if it is necessary, the animal models for experimentation, or you can supply it.
Ah, okay.
In
I'm
vitro
very
or
sorry.
ex
Now
vivo.
I heard. I'm very
sorry.
sorry. Yes. Animal models are still necessary for development of some pharmaceutical, of some medicines. Now I do believe animal models are completely unnecessary for development of cosmetic products because, well, you have already alternative in vitro methods for development of cosmetics because they're not supposed to exert a systemic action. They're supposed to exert some localized action in the skin. So these in vitro models can fulfill these demands for cosmetics. That's 100%. You can abolish animal experimentation in cosmetics. But for medicines, you can still not. I do believe in the future we will be able to abolish animal experimentation up to a point we can have clinical experiments, but nowadays we can't. There's some pharmacokinetics. You need an animal model before you go to humans because the risk, you cannot afford the risk of having something unpredicted to a human volunteer. So you need this animal model. But I do believe we will get there, and we have to use as maximum our in vitro models as much as possible. So when it's not possible anymore, and the next step would go to clinical, then this step you may need animal. But until then, I'm against, and I think we can reduce a lot.
Hi, doctor. Hi, can you hear me?
Mm-hmm.
My question is, what kind of materials do you use or have you find useful in your ophthalmological models? Like for your nanoparticles, what kind of materials do you use in your nanoparticles to apply or to for drug delivery in your model?
What types of materials? Well, we have worked with liposomes, so we have phospholipids, and we have worked with nanolipid carriers, also solid and liquid lipids. We have some studies with metal nanoparticles, so we have some studies with iron nanoparticles that had some interesting results. Now we're working with some metallic nanoparticles, so we're investigating some uses of oxide cobre, yeah, and nanoparticles and diverse material. Did I answer your question?
Yes, but what are lipid nanoparticles really stable for drug delivery in the eye? In the eyes, in the ophthalmological model?
Yes, the nanostructured lipid carriers. Then you have a mix of solid lipid and liquid lipid.
Thank you.
Are you hearing me? Do you hear me?
Yes.
Yes. Thank you for your lecture. I enjoyed your lecture this moment. I have a couple of questions. The first one is that your lab is near to the Lauter house.
I didn't understand.
Okay.
Sorry.
Your lab is near to the Lauter house.
My lab is near to the slaughterhouse. No,
No.
it's about 1 hour distance.
Okay. This
Yes.
this question is about how long time did you use the tissue? For example, the vaginal of the pigs in your lab for testing.
Yeah, it's about 1 hour distance. So we maintain them in these boxes with the thermal boxes, not in direct contact with ice. We protect them from the direct contact with ice, but these thermal boxes, and we transport them like this. And it's enough, just maintaining this low temperature. But for this mucosal tissues, vagina, buccal, and ocular, when you get to the lab, you start immediately the experiment. You just need to leave everything prepared. And sometimes my students stay in the lab until 1 a.m., 2 a.m.
Yeah.
until they're finished the experiments. It's very laborious.
Yeah.
It's very hard.
In the same day.
Yeah, the same day.
Okay. I have a doubt about your model, your prototype of the ophthalmics. What kind of layer or what kind of material is the layer that you use as model?
Okay. Well, we have tested some multiple types of materials, but we have tested some polymers, plastifiers, and gelatin with some plastifier, and we have tried poloxamer, some polymer, plastifier, and mucin. So we use mucin mixtured with the polymer, so we can have this mucoadhesive behavior. This is one type of material we're thinking about, but in the next development, we're trying to make some in situ polymerization. We're thinking about making a synthesis that we covalently bound the mucin to the polymer, and then we can polymerize this before the experiment in that prototype material, so we can have something more like the cornea surface. So we have this basic liner now with mucin, but we're still working on that to develop it more and more to have it a more resemblance to the cornea surface.
Okay. Finally, what do you think about the in chemical models? Did you know that?
The chemical, which one?
In chemical, you know, we use in vivo, in vitro, in situ, in silico, but other models are in chemical.
Well, yes, they can be used. I think maybe in a previous phase, when you're selecting when you're selecting molecules, I think when you're maybe, I don't know, I don't know if you agree, but I believe they would be more useful when selecting molecules and materials. But once you have
Oh, we
Hello?
we. Do you hear me?
Now I'm back.
Do you hear me?
Mhm.
Oh, no, thank you very much for your lecture. Muito obrigado por the palestra.
Muchas gracias. Muchas gracias. Es un placer.
Hi. Hi. Do you hear me? Hi. Hi.
No, I don't hear now.
Hi. Do you hear me?
Yes.
Hi. Thank you for your presentation. I have a doubt. If I make a new product and use organical like cannabis, what is the advice or advantage that I apply in this model in vitro? What is the really challenge to use this cannabis to apply in skincare or in new products oriented to cosmetic take care of the skin?
The challenge.
Yes, to use cannabis or include cannabis, that is a molecule that have a lot of don't have stability.
Oh, sorry, I can't hear.
Don't worry. My question is, if I want to make a new product and I take cannabis, what is the advice that you give us to maintain the stability or the things that cannabis produce in the skin?
You want a product for which action?
Skincare and use cannabis in the campaign.
Mm-hmm. Well, the main challenge, you cannot discard the sensorial feeling of the formulation. You need to maintain the active stable, but you also need to think about the sensorial feeling. For this purpose, the formulation must be pleasant to use, and not sticky, and not to be excessive oily. I don't know if I answered your question.
Yes, thank you.
¿Alguien más tuviera alguna pregunta o comentario? Si no fuera así, doctor Thais, ¿quiere hacer la última pregunta?
Thanks so much. Can you hear me?
Yes.
Yeah. Okay. This about your formulation model. Do you recommend that test the two ways for formulation assay? You recommend say the horizontal position and vertical position in the same in the same measure?
Do I recommend the vertical position?
An horizontal one.
I don't understand. I'm
Yes.
sorry.
When you test your model for formulation,
Mhm.
you test
Mhm.
in two ways, in vertical and horizontal position.
Yes.
Did you recommend apply both of them?
Well, it depends. It depends on the use you're thinking about. Because Depends on the region of the body the formulation will be applied to. For this example, we intend to apply in the eyebrow. But if you want something to just, okay, it's a skincare, I'll apply to my whole face. I don't see the necessity of using the vertical model. I think just the horizontal model would be enough. So this leaking, this dropping off, it's not critical for your action. You're just going to spread everything to your face. So it's not important. It doesn't make a difference if it's horizontal or vertical. It depends on the specific goal you're trying to reach, I guess.
Thanks so much.
Bueno, estamos ahora al final de nuestra sesión del Seminario del Departamento de Farmacia. Dear Dr. Thais Gateri, thanks a lot for your time and this interesting topic. We are now at the end of this presentation. So, muchísimas gracias, Dr. Thais Gateri. Thanks a lot, and I will send you by mail your certificate of participation.
Thank you a lot. I would be very happy to welcome any of you here. We're in a difficult time in Brazil right now. We're hoping for things to get better so we can have more finance for our research.
It's good.
And I'm hopeful to maybe host some of you someday. Who knows?
Thanks a lot, and have a great weekend and good afternoon there. Thanks a lot.
Thank you for the invitation. Thank you.
Thanks. Muchísimas gracias a todos y muy buena tarde.
Bye.
See you.